Good afternoon. My name is Vadim Zhernovkov. I'm a program director of the MSc Artificial Intelligence for Medicine and Medical Research. And today we are going to talk about this master program. We will cover different aspects, but first of all, I just want to say that please put your questions into the Q&A area. Just click on the button and just you can enter your questions. And you can write your question during the presentation, and if we will have time, we will discuss these questions. Or, for instance, maybe we can postpone to the end of our sessions. And first of all, his agenda. And Professor Walter Kolch will talk about AI and medicine and drug development. Then I will introduce the course, some structure, requirements, and then we will talk about our models. And our model coordinators will present their models. And first of all, Walter Kolch is a director of systems biology Ireland at the University College Dublin, and he originally trained in medicine at the University of Vienna, and later he worked in the pharmaceutical industry, research institutes, and academia across several countries, and he leads multiple research projects focused on cancer research, and he's recognized internationally for his work in precision oncology and systems biology, and he's a member of the Royal Irish Academy. Walter, can you share your slides?
Yep. Okay, can you see the slides?
But this is okay. Now it's great. Yeah.
Yeah. Okay, Grant. So thanks for the introduction. Welcome everybody to this information session. I'm just going to kick off by telling you a little bit about where we use artificial intelligence in medicine and in medical research. If you think about what we are doing with artificial intelligence, it's really sort of a digital scalpel that helps us to understand medical and biomedical data and really puts medicine and biomedical research into the modern information age. So it's a tool, it's a very powerful tool which we can use and which is fairly new and also very exciting. So if you look back a little bit, about 20 years ago, the first human genome was sequenced. And this is also coincidental with when we put the beginnings of personalized medicine. And this is actually not a coincidence, but genome sequencing really opened the door to personalized medicine and to precision medicine. I'm using personalized medicine and precision medicine meaning as exactly the same term. So some people say personalized medicine, some people say precision medicine, but it's meaning exactly the same thing. And sequencing the human genome really was the key event. But the human genome is a huge amount of data. It's 3 billion data points. And we have to deal with this, and we have to analyze this, and we have to understand these data points. And this is tricky, but if we can do this, genome sequencing has a huge impact and a huge benefit for patients. So this is just one example. And this is cancer patients who were treated according to their genome sequence and choice of drugs tailored to exactly their mutations in the tumor. And if you can see here on the survival curves, the blue curves is where the patients could be matched based on their mutation to a personalized therapy. And the orange curve is where patients could not be matched, either usually because there was no drug available which could address their mutations. But you can see this huge difference in survival. It's actually a 4-fold difference. So if we can genome sequence and we can design a personalized therapy for patients, the survival chances change dramatically. So this is just one example for how powerful this is. But it also shows how important it is to interpret data. And this is not only confined to genome sequencing. We now have a lot of different omics technologies like metabolomics, proteomics, imaging, transcriptomics. and many other data which we can generate. And we are very good now in generating data. It's very fast, large volumes of data, but we still need to interpret them. And this is now becoming the bottleneck. So it's no surprise that Harvard Business Review puts data scientist as the sexiest job of the 21st century. And this is across all domains, including healthcare, including biomedical research. So if you look at who is using AI, so it's the top companies in the world. For instance, the data companies like IBM, Google's, Microsoft, but also top medical research. So the New England Journal of Medicine is the top medical journal in the world, but it's now penetrating the whole sector. And also, if you look at the World Economic Forum, it's real, the global economy. So AI is everywhere. And in the future, AI will be even more everywhere. So this is just the growth of AI applications just in the healthcare market. And you see it almost went up by tenfold between 2014 and 2021. And this trend is going to continue. So AI is really going to grow like crazy. And if we look at what AI has been used for, in the last decade, it was mainly medical products like equipment, hardware. Nowadays, we are using it for interpreting data which come from variables, the big data which come from omics, for health analytics. And in the future, we will increasingly use it for robotics, like robotic surgery, augmented reality. And we also will use it more to focus on preventative care, which is becoming more important in healthcare. So what we are seeing now at the moment is actually that biology and medicine are becoming data sciences. And the emphasis is really shifting from the production of the data to the interpretation of the data. And so what we're going to be using AI in medicine is several areas. I just pick out 4 important ones, and these are also areas which you will hear about in the MSc course. So one is image analysis, one is precision or personalized medicine, another one is drug discovery and drug development, and then the ultimate aim is constructing digital twins of patients, which is computational avatars of patients where we can simulate their disease, but we also can simulate interventions and then design the best possible intervention on the computer before we actually give it to the patient. So I just give you very quick examples. The first one is imaging, and this is already where AI makes a big difference in the clinic. So the first paper on AI being used for diagnostics in imaging was in 2017, and it showed actually that AI could diagnose skin lesions equally well as the best pathologist, and that in the meantime, AI is actually better than the human pathologists. And the advantage is, of course, AI can work 24/7 and pathologists cannot. Another big advance was retina scans. So this is where they take a picture of the back of your eye, and what you can see here are all these blood vessels. And depending on the morphology of these blood vessels, you actually can tell whether a person is likely to get diabetes and is likely to get hypertension. And AI actually can do this much better than humans. So we can anticipate who is at risk getting diabetes or hypertension and intervene early, which is very important for the prognosis. Then it's also being used for screening. So there's about, in the UK alone, about 100 million mammography pictures taken per year. And this is way too many radiologists can look at. So AI is being used to actually sort out the normal ones so that the pathologist only need to look at the suspicious ones, and that cuts it down by 99.9%. So huge saving in terms of work. It also has been used to diagnose COVID or who is at risk of developing severe complications by COVID. And it really turns out that this is going to be now clinical decision support systems for the doctor, where the doctor can use AI to get a recommendation or improve their diagnostic abilities to tell what's wrong with the patient. Another growing area is that of precision and personalized medicine. And so we now see that AI is being used to optimize cancer treatments, to predict cardiovascular risk, to manage diabetes, to diagnose rare diseases, immunotherapy. So from this list, you already see that it has growing applications in areas which are very, very important in medicine. And last but not least, we also use increasingly AI in pharma industry. So for diagnostics, for drug discovery, then developing the drugs, but also then producing the drugs. And there is a prediction that by 2030, more than 50% of the drugs will have been designed and produced using AI approaches. Currently, it's about 160 to 170 drugs, but this will change and it will change upwards. So there's really big prospects for AI also in the pharmaceutical industry. And these are all these things which you will learn about in the course, which Vadim will explain how the structure is. So I'll stop here. And if you have any questions, please put it in the chat. Sorry, in the Q&A.
Thank you. Thank you, Walter. Can you see my slides? Can you see my slides?
Yeah, I can see it.
Great. Yeah. Thank you, Walter. I will talk about the program structure, different models. And no, first of all, just want to say that this program was developed by Professor Walter Kolch 4 years ago, Professor Walter Kolch and his team. And the idea of this program that if we prepare specialist with mass and stat background and or biological background that are able to interpret data. For instance, currently we have a lot of different type of data. It could be gene expression data, protein expression from mass spectrometry data, or from alternative platforms. It could be images from MRI scans or from pathology slides, all from clinical data. But as Walter mentioned, one of the bottleneck, one of the challenge is interpretation. And one of the question, how we can use this multiple type of data, a huge amount of data to improve drug development, to develop new drugs, and how we can use this data to increase diagnostics, clinical diagnostics, and introduce precision or personal diagnostic for our patients. And this course offers different models with covering state-of-the-art methods in AI and machine learning and the application for biological and medical data programming, mainly Python and R, and data visualization, and also a lot of different medical-oriented courses who is focusing on different aspects like clinical trials, statistics for clinical trials, and so on. And I would highlight that the course has been developed and led by a multidisciplinary team from Systems Biology Island and UCD, with extensive experience in collaborative research project between academia and industry. And team members have experience in R&D project in collaboration with biopharmaceutical companies. And first of all, who is this course for? First of all, this course could be interesting for computer scientists, data scientists, and that's why when we organize an interview, I usually ask linear algebra, calculus, probability theory. This is quite important for us. Also, we offer this course for biologists or for medical specialists, clinicians but one of the important criteria is good programming skills. We require R or Python. And our general requirements: bachelor degree, master background, or molecular biology or medicine, and English test for international students. And here's a list of our courses. We offer the most popular course is a master course. This is 1 year course, and this is 90 credits. And we will start in September and finish in middle of August. Next, we also offer part-time course. This is 2 years, same structure, same idea, same models, but you can take different models during 2 years. And also we offer graduate diploma, this is 9 months. This is same as master program, but we did not include to the course our summer models. And this is a 6 credits model. And I would highlight that first of all, this is in-person course. You have to be on campus at least 2-3 days per week. And we deliver our course in hybrid model. Some of models could be delivered completely 100% in online. But for instance, our core models are delivered only in person. And during this presentation, during this information session, we will cover mainly core models, but just to give you a sense in terms of structure, in terms of what we will learn. First of all, we will have 3 trimesters, autumn, spring, and summer, and we will have core models and optional models. Core models mandatory, optional models can give you some flexibility. You can choose any optional models just to improve your programming skills, for instance, in Python or R, and to improve your machine learning. And also we offer clinical trials related or medical related models, and also offer introduction to biostatistics. And this introduction to biostatistics mainly about some statistics in biology, what is t-test, normal distribution, how we can compare different samples, different conditions, what is ANOVA test, what's the difference between parametric, non-parametric tests, and so on. Next, spring trimester offers more specialization, and we offer more specialized models in machine learning, for instance, deep learning. And as part of this model, our students learn deep learning algorithms, neural networks, and the implementation using different tasks, different projects. Also, network analysis. Biology actually is a network. A lot of different network-based tasks like protein-protein interactions, patients, similarities networks, and so on. And that's why for us, it is important to know some methods using network-based analysis. We also offer information visualization. Definitely, it's quite important. And bioinformatics. Bioinformatics model mainly offer some structure biology, mainly focuses on structure biology area. And then summer trimester offers specialization like AI for personalized medicine, AI for medical image analysis, and AI in digital pathology. And these core models, we will talk about these core models during this information session. I would like to show some slides about research internship. This model has limited capacity and entry is competitive. And first of all, the aim of this model is to provide hands-on experience in data analysis using real-life projects in collaboration with our collaborators, with our supervisors from academia, clinical laboratories, and industry partners. And we launched this model 2-3 years ago. We tested different strategies. We have been working with academia partners, with external, for instance, we tested a strategy when we worked with one of the university from USA, from the University of California, San Francisco. We also tested some strategy with industry. And currently we offer 6-month internship. This is non-paid internship. And usually supervisors meet with students every week, one meeting per week, and discuss some research progress. And criteria for selection to this include students' performance in the autumn trimester and the initial background. It could be some, if you have a good master background, you definitely will increase the chance for selection. And we work with academia, with some clinical partners. Like, for instance, this year we work with St. Vincent Hospital. And here's a list of internship project for this year. Some of the project currently we offer more than 11 projects this year. And for instance, one of the projects, the first project is analysis for single cell transcriptomics data and spatial imaging to uncover a mechanism of rejection in kidney transplant from St. Vincent Hospital. And Dr. Andrew Malone supervised one of our students. Also, we offer this year machine learning for CRISPR genome editing and RNA therapeutics. And Rory Johnson leads this project, and have one student that is dedicated to this project. We also work with image-based analysis together with Fiona Friedman, and also I supervised, for instance, personally 2 students. One of the students who works with foundation models, and we focus on T cells, T cell biology, and we try to create a foundation model for T cells. That then the model should be used for some downstream analysis, like inferring gene regulatory networks or some confirmation of checking some hypotheses. And the second project is multi-omics integration, and we use different type of omics like DNA-seq, RNA-seq, spatial transcriptomics, and we use CatBoost or some graph-based method for integration multi-omics to understand heterogeneity of microenvironment in intrahepatic cholangiocarcinoma cancer. And also, I would like to show also one of the projects that David Hughes from Conway Institute leads this project, and this is about gut microbiome and how and analysis in terms of pancreatic cancer. And I would also like to show some example of inter-school collaboration from the last year. And one of our students, Kevin Whelan, worked with this project, and this is kind of example of collaboration between different schools, between School of Medicine and Computer Science. And Kevin developed one of the algorithms using generative AI and generated in silico images for ultrasound images for modeling. And his kind of advice, first of all, if you are part of our program, I would suggest to apply for fellowships. We have really good some kind of example of experience with this with our students. Also participate in conferences, participate in biohackathons. There are a lot of biohackathons in Europe. Just apply and publish, and definitely it would be good if you can publish your research projects. And in terms of career prospects, our career, based on our graduates, what we see now is that we can highlight different maybe paths. First of all, it could be maybe data scientist or machine learning engineer in industry. I also would like to suggest to consider graduate programs. A lot of different companies like AstraZeneca or Novartis offer graduate programs for machine learning specialists, and also PhD programs. And here is the example of some open positions from last year, from Novartis, from Optum. And regarding PhD, our graduates were quite successful. And for instance, last year they joined top laboratories from USA or from Europe, and we also have a lot of local students from in ECD or University of Limerick. And to apply, you need to provide some documents. I would suggest to prepare. We usually require CV and short motivation letter, transcripts, reference letter, and English test for international students. And yeah, we will discuss later at the end of the presentation regarding recommendations in terms of some courses that could be useful. Now, I will stop my sharing. And if you have some questions. Yeah, we have some questions, but we can discuss later because we do not have enough time, maybe. Yeah. And next presentation will be from Aleksandar Krstic and Luis Iglesias Martinez. Aleksandar has a assistant professor position and at School of Medicine, and Luis is a research fellow at Systems Biology Ireland. Aleksandar, can you?
I'm sharing. Can you see the presentation?
Yes.
Excellent. Thank you very much, Vadim. Good day, everyone. So, as Vadim already said, Luis Iglesias Martinez and myself are part of one of the core modules that you have in the autumn semester. The official name of this module is Biological Principles of Cellular Organization, and we tend to call it Crash Course in Biology. So, just a second. So let me introduce briefly first myself. So my name is Aleksandar Krstic. I'm originally from Serbia, and I'm currently employed as assistant professor, Dastra fellow at UCD School of Medicine, and conduct my research in Systems Biology Ireland as a group leader. I work with a team of researchers that include classical molecular biologist. I have a PhD student who is a clinician, and also work with, as we call them, computational biologists, dry lab people. So my PhD was obtained in Systems Biology Ireland at UCD many years ago, and my original studies, bachelor and master's course, was done in Serbia, in Belgrade, primarily in molecular biology and physiology and genetic engineering. So as you can see, I will not say how many years in science, but a lot of different types of research. So if I could briefly tell you about my interest in science, is primarily cancer. My work is focused at the moment on colorectal cancer, specifically aggressive form of early onset colorectal cancer that affects young population up to the age of 50. And neuroblastoma, pediatric cancer that affects very young population and infants mostly up to the age of 2. If I can summarize all of my research in 2 words, that would be gene regulation. I'm interested in genes, how genes work, what regulates them, how they talk to each other, how this regulate processes in the cell and in health, and also in different disorders. So, Luis, would you mind introducing yourself?
Hi, everyone. I'm Luis Iglesias Martinez. I'm a research fellow in Systems Biology Ireland as well. I lead a group where we study AI and developing applications for cancer genetics, and I'm a principal investigator of the Magic Eye project. I have my PhD, I did it here in UCD, but I did a master's in biotechnology in Chalmers University of Technology in Sweden, and I'm originally from Mexico, where I got a degree in chemical engineering. My main areas of interest are AI in genetics and childhood cancers. Thanks, Alex.
Thank you, Luis. So let's go with the formal title of this module and explain you what are our teaching objectives and what is the aim of this course. It's a core module, and it's primarily aimed at computer science with little to no biology background. Some of you had questions, and Vadim reiterated that. So this module is aimed for people from different backgrounds, computer science. We had students from neurolinguistics, from physics, with PhDs in computer science, people who work in companies on face recognition, already in some fields of AI, some fields of the statistics. So very, very, very different backgrounds. One important thing is that we are aware that some of the people with biological background are interested in this field, in AI, and we cater for these students. So this is not the module that we want you to feel bored. We want you to feel engaged and to have acquired the knowledge that would be useful to you. So the content of this module goes from very basic concept that you will that you acquired in your high school. For some of you, this would be a refresher, and it slowly accelerates and goes to very complex biological questions in health and also in different disorders. Specifically, cancer is one of the main fields that we would cover. So this is a foundation. We are trying to give you understanding of the complex biology in health and in disease. And the module has two main parts, and Luis will explain the second part of the module, but generally we want to have a holistic approach that is required in AI combined with biomedical and health sciences, that you can apply AI, you can apply machine learning in genetics and molecular biology data sets. So the approach is going from very, as I already said, from very simple to highly complex ideas and concepts. We tend to divide this module into two units. The first part is delivered by me, and some of the scientists that work in Systems Biology Ireland or at UCD Conway Institute or School of Medicine. And we start from basic concepts of cells and tissues. Then we go to DNA, RNA, and proteins as the building blocks of the cells, and the basic concept of genetics and epigenetic regulation. Then we go to more complex ideas like how cells communicate with each other and within the cell, that we call cell signaling, and acquire the basic concepts of how some of the targeted therapies and drug mechanisms work. And of course, the important part is cancer biology. So as you can see, this almost looks like some of your biology courses that you had in high school or college, but to keep this content relevant, we strongly encourage you to dedicate time for self-study, for self-development, and we are very open, which is done through your reflective pieces that are parts of the course, that you can embrace any topic that interests you in the field of biology or biomedical sciences. So the students in previous years were very happy to write short essays about the origin of life, the origin of mitochondria, role of genetics in psychiatric conditions, and many, many other topics that are not part of this, strictly part of this course, but are relevant and interesting. So engaged and self-directed study that will help you to embrace all of these ideas. From the first part, we move to the second part, and I'll hand over to Luis to explain.
Thanks, Alex. So in the second unit of the course, what we want is for the students to be able to apply the theory that they've learned from biology into real-life data sets and to use some of the algorithms that we commonly use to analyze genetic and molecular biology data sets. So some of the things that we teach some basic bioinformatics, pathway enrichment. So this is finding patterns in gene expression that tell us what signaling cascades and what proteins might be driving certain phenotypes of certain disease. We also do some data analysis for these kind of biological data sets and survival analysis. And what we want is for the students to be able to interpret the results and to be able to use what they learned from the theory of biology and cellular organizations to use what they learned in a real life data set and to be able to conduct a basic data analysis workflow. Sorry, yeah. So why is this module important? What we want is to be able to teach students to understand cell function and the basis of disease, and based on it to be able to design and build powerful AI applications on the line. So we want to give you the context so that you can think of important problems and think of AI or machine learning approaches that might be able to, at the end, change patient outcome. So that's what we want that is why we think this module is important. This is a core module, so it's not an elective module. And the reason for this is it is meant to provide you the foundation to understand and embrace biological and medical data. So this is meant to be like a module that you take at the beginning of the master's, and it sets you up for the following modules. So just a little bit on delivery. So this is an in-person module. It's more or less 2 hours a week in lectures, but there's also individual and autonomous learning work. And it is a mix of lessons and tutorials. So you'll have taught lessons with either me or Alex, and then we'll take you through different tutorials in which you will be programming and analyzing real-life data sets. And we want the module to be to have a lot of active participation, for it to be a place where you can engage and discuss your ideas with your peers and with us. And we're always happy to discuss research project questions with you. So thank you very much for listening, and look forward to seeing you when the master's begins.
Thank you, Luis. Thank you. And moving on, our next presenter is Melinda Halasz. She's an assistant professor at School of Medicine, and her specialization is cancer research. And Melinda, you're welcome.
Thank you, Vadim. Okay, good afternoon, everyone. My name is Melinda Halasz, and I'm an assistant professor in pathology in the School of Medicine. And I'm coordinating the Precision Oncology module, which is offered to the MSc in AI and MSc in Precision Medicine programs. So in this module, you will have lectures around four major topics. At first, we will briefly review cancer biology concepts. Then we will discuss cancer genomics with bioinformaticians. And then the third block of lectures will be delivered by clinicians, pharmacologists in the space of molecular oncology. So you will learn about what is available in the Irish hospitals in terms of precision oncology. And then in the fourth lecture block, you will learn about how mutations will affect signaling networks. So in this module, hopefully by the end of this module, you will appreciate a multidisciplinary approach to patient care, and you will learn about what is the diagnostic workup for a cancer patient. You will learn more about the role of pathologist and bioinformatician in making a precise diagnosis. And also you will see how a precise diagnosis will affect the deciding on the proper treatment for the patient. In addition to what available in the clinic in Irish hospitals, you will also develop an interdisciplinary, multidisciplinary mindset, which will aid you to understand precision oncology research, and you will be able to translate it into the clinic. So we will focus a little bit on cancer genomics and other omics technologies, and also you will have many lectures given by clinicians and also researchers, and also you will appreciate the perspective of the patient as well. In terms of delivery, you will have 2 lectures every week, and for each lecture block you will have a tutorial as well. And because you might have very different backgrounds, you will have mainly group assignments as assessment. We will assign you to groups to make sure that every group will have a computational and biology background. And the 2 main projects you will work on is, one, you will be given a molecular pathology report, which at the first tutorial together we will interpret. and then you have to come up with a patient case. So you have to appreciate the multidisciplinary team meetings which occur in the clinics. So each of you will be assigned a role, like surgeon, radiologist, oncologist, and so on. One of you will be the patient. So you will present the patient case. This presentation will be after the third lecture block, once you had all the lectures on the most common cancer types: breast, lung, colon cancer. Then your final and biggest assignment is a group project in the space of precision oncology, where, depending on the skill set in your group, you will come up with a question and you will analyze some publicly available data sets, and you will provide some new insight into a precision oncology question. And that's it for me. Thank you, and looking forward to meeting you in person in spring 2026.
Thank you. Thank you, Melinda. And our next we are going talking about Oleksii Rukhlenko model. Dr. Oleksii Rukhlenko is an associate professor at School of Medicine, and his specialization is cancer research, modeling, machine learning, AI. Oleksii,
Hello, everyone. Can you hear me? Can you see my slides?
Yes, we can.
Okay, excellent. So, good afternoon, everyone. My name is Oleksii Rukhlenko, and I lead the summer module, which is called AI for Personalized Medicine. So, a brief background about myself. The main question which I try to address in my research is how signaling networks inside the cells shape the landscape of cell fate decisions. So basically, in simple terms, how cells compute their fates. And I try to answer this from different points. My main expertise is computational, mathematical modeling, machine learning, partially control engineering, but I also try to acquire expertise, use knowledge from different areas. We have wet lab parts and experiments being done. We try to introduce some knowledge from structural biology. So this is all we try to work and connect together to obtain more or less holistic picture. And yes, this is the main idea, to obtain holistic picture when the model is so good and so detailed. So it's kind of digital twin of physical object, like in computing, like in machine engineering, we have a digital twin of turbines, airplanes, spaceships. We try to build a digital twin of a human. So, and now what is the bridge between the course which I teach and my own research is exactly the piece where such digital twins can be potentially used to personalize, to understand personalized predictions, either based on what optimal treatment should be done, or at least what should be expected in terms of prognosis. So we try to understand how, in terms of course, we try to open a bit a field for you, how based on network knowledge, how based on machine learning knowledge, we can understand what will be the prognosis for each specific patient for each specific treatment. So yes, the main idea behind all the course is we heavily rely on survival analysis. Most of the class will be using different approaches to survival analysis. So the main idea is to analyze patient data. When we have the data about patient survival and their molecular data, and the main aim of my project is to make you able to analyze together molecular omics data like PPA proteomics, transcriptomics, genomics, together with clinical data, and integrate different layers of molecular data tied up to survival data. So survival data has a little bit its own specifics. Most of methods are usually based on discrete things like classification, or continuous like regression. In this case, we use both. So usually people a little bit struggle to understand that we actually have to use both things simultaneously, and this is not something that usually done in other fields. Usually it's either or. But in this case, this is the key. We need to analyze simultaneously both classification and regression for patients and molecular data. And okay, in terms of methods, we will cover different types of methods, from the most simple ones like feature selection to neural nets to some mechanistic-based methods. So here I will try to analyze more deeply the dichotomy between knowledge-based computational work, which is usually called modeling, and data-driven computational work, which is usually called machine learning. These are quite approximate names because in modeling we use some elements of machine learning and vice versa. So we will try to understand how we can bridge these things to increase the robustness of modeling. So in terms of timelines, there will be 2 personal assignments and 1 group assignment. So personal assignments will be applying these different methods to different data sets. The group assignments, I will try to provide you as much as possible different methods, and it will be up to you to show how you can use them together. During all time until your last time of submission and presentation, will be working in person with you, looking into your data sets, into your code, helping you to finish your work. So until the exact deadline of submissions, I'm fully yours to help you with. Usually this course requires a lot of person-to-person sitting and person-to-person discussing of your own project. And I will, in this case, all this time during this course, all my time is yours to help you to work with this. And in terms of data, we will also learn how to use absolutely different databases, which contain data about cell lines, about publicly available patient data, about different layers of publicly available patient data, like genomics, transcriptomics, proteomics. And we will learn how to basically integrate them together to obtain more or less holistic picture, and based on this picture, predict outcome of different treatments for specific patients. So yeah, this is it, and I'm happy to address any questions if they arise.
Thank you. Thank you, Oleksii. And if you will have questions, just please write in the Q&A area. And our next presenter will be Dr. Eoin Brennan, and he will talk about high throughput technologies model.
Thanks, Vadim. Let me just maximize this. Can you see my slides and hear me okay?
Yes, we can see. Yeah.
Great. Hi, everyone. So thanks for the invitation today. So as Vadim said, Eoin Brennan is my name. I'm an assistant professor in the School of Medicine. And I teach this particular module, high throughput technologies, which is delivered early in the program in autumn, 5-credit module. And before I tell you about the module, just to let you know who I am, based in the School of Medicine, the Diabetes Research Center. I have an interest in kind of vascular complications of diabetes, cardiovascular disease. Very much a wet lab scientist by nature, so we work a lot with human cells and tissues and work with consultants and so on, surgeons, getting access to tissue, trying to understand the pathways that change in healthy versus diseased cells and tissues. And to do a lot of that, we have to use a lot of technologies, which is why I like this particular module that I teach. And it complements very much the module delivered by Aleksandar and Luis in the program as well. And so with this high throughput technologies module, we look at the human cell or the human tissue and the layers of information that's contained within them. So we talk a lot about the genome, the transcriptome, the proteome, and the metabolome. And the way we try to do it is we will cover the fundamentals, a little bit of the biology to overlap somewhat with what you're getting from Aleksandar and Luis. And then on top of that, then we talk about, okay, if we have this appreciation for your DNA or your proteins or your metabolites, how do we go about measuring them? So with some of the technologies that are available for us to do that. And again, you've seen slides like this already, but again, this is kind of fundamental to the general setup of this particular module. or as we move through the module and the sessions, we look at different layers of biological data and think about how we might build a picture, and which is obviously the focus of precision medicine and personalized medicine, is to get that full picture of biological data coming out of a cell or a tissue. And when you have all that data and all that information, perhaps then you can start to think about new therapies, thinking of better therapeutic outcomes and so on. So we work through these individual layers slowly but surely throughout the module. And the aims of the module, again, as I said, we're going to talk about kind of the fundamentals first of all. So I don't expect people to come in having lots of biology. In fact, some students have very limited biology coming in, and we work through it fine. And so it's not an issue for us. We talk about the basic principles of these different biological layers. And on top of that, then with these sessions, we talk about some of the technologies that are available to us to actually investigate some of these things. How would you measure DNA, RNA, and protein, and so on? Some of the limitations and advantages of these. What technology would you use for your particular study? How it's delivered then, so as I said, it's a 5-credit module in autumn. Our sessions are weekly. I think it's 2 hours per session. And you can see here the actual titles. We talk about the human genome, talk a little bit about the transcriptome, sequencing technologies, and epigenome. You get those from me. So the first half of the module is primarily delivered by me. And then I'm very happy to invite in experts within our university who talk about various different topics. So we have, for example, flow cytometry. We've got Professor Alfonso Blanco, who directs the core facility. For proteomics, we've got Professor Matthias Willem, who directs the proteomics core. And towards the end, we'll talk a lot about metabolomic principles and technologies with Professor Brennan, who heads up the core facility at UCD. So it's a nice introduction at the start to the fundamentals. And as we work through the module, we will talk about different types of biological data and get the relevant experts in then to give you that information. How it's assessed then, very briefly, just to give you an idea. 50% of it in the bottom is just a traditional exam, a multiple choice exam that you get towards the end of the module. And the other 50% then is made up of group work. In particular, a short journal club presentation that you work in assigned groups to present on a particular paper. And then we've got 30% then, which is towards discussion forums, which is a new form of assessment I've introduced in the last couple of years, which students actually really enjoy, where you work as a group to just chat about a particular paper that you can pick as a group, talk about the pros and cons. You can agree, you can disagree. It doesn't really matter. The whole point is that you just work your way through it and give your opinions on that type of paper. And again, to give you an idea, even last year, what we did with the class with some of this stuff, just to give you an idea of some of the content we covered for discussion forums, which can be done online on your groups. Some of the topics: ethics of blind DNA, perspectives on who owns your genomic data, the role of religion and spirituality in genetics. That's actually quite a popular one, believe it or not. Precision nutrition, and so on and so forth. So a little bit different, a little bit abstract, just to get you thinking and get you discussing about your own opinions on some of these topics. Okay. So I think that's pretty much it for me. I want to thank you for listening. You're welcome to ask questions toward the end, and I look forward to meeting hopefully some of you later on in the year. Thank you very much.
Thank you, Eoin. Our next speaker is Kathleen Curran, and she will talk about AI in images, AI for image analysis. Kathleen, can you show your...
Thanks, Vadim. Yeah. So thank you for the invitation, Vadim. Okay, you should be able to see my screen now. Okay, can you? So my name is Kathleen Curran, and I'm an associate professor in the School of Medicine with a PhD in computer science. So I'm teaching this module from the perspective of having a medical imaging understanding and also technical background and expertise. And we'll also have the support of a couple of tutors who will help with the practical Fang and Satang. So I'll just go through the module content and outline some of the practical sessions that we do and the assessment. So this is quite broad in the sense that we'll start with an introduction to medical imaging, and AI. And then, so everything sort of from acquisition to pre-processing, post-processing, so some traditional medical image analysis techniques, also like feature extraction. And then we'll move on to traditional machine learning techniques, and then on to deep learning. So covering the more advanced and recent topics like foundation models, etc., but also our CNNs and UNet and NNUNet and different types of deep learning models. Very much focus on the application of these techniques to medical imaging. So exploring everything from specific applications, including computer-aided diagnosis, image segmentation, or defining regions of interest, or outlining boundaries of specific features in the image that are of interest to clinicians. Also disease classification, disease detection, prognosis, etc. We'll briefly consider some of the ethical and regulatory issues in AI. And a good focus of the module will be around interpretability and explainability. So being able to demonstrate where the model is focusing, why it's focusing there, look at fairness in AI, bias in some of the training of the models, etc. And the clinical implementation and evaluation of AI. So with that in mind, I guess we'll have a lot of invited speakers, invited guest lecturers, including clinicians and including world's leading experts. The module will be delivered in person, but we will have a couple of Zoom lectures. But it is essential that the students are in person for all the sessions, because we'll have some Q&A at the end of lectures also, and it'll be very interactive. So finally then, sort of leaning towards future directions in AI medical imaging, large language models, and some of the more recent trends. In terms of the practical, so we will look at using Google Colab, working with open source data. So we'll have some introductory practical sessions on how to use Google Colab, setting it up, running AI models specifically for medical imaging tasks. And we're focusing specifically on segmentation and classification. So learning how to access and pre-process publicly available data, downloading the data sets, interrogating the data sets, becoming familiar with the annotations, and looking then in particular at data labeling, so how to annotate medical images with labels for training supervised learning models, exploring different annotation tools also, for example, like 3D Slicer, Supervisely. Azarix is an open source tool that a lot of clinicians use, although they will not work with the free version that we will work with. Exploring other techniques for quality control and annotation consistency, inter-rater agreement, etc. So we'll also, in terms of the practicals, there will be another session on data augmentation techniques, so how to increase, say, the size of your training data by applying like rotation, intensity changes, flipping, etc. to the images. There'll be another session on interpretability and visualization of the segmentation results. So learning how to visualize and interpret the segmentation results using different tools, exploring different techniques for overlaying the segmentations of medical images, whether it's color coding or contour lines, etc., and how to assess the quality of the segmentations using different metrics, and also domain knowledge from an explainability and interpretability point of view. We look at transfer learning, learning how to use different transfer learning techniques to adapt pre-trained convolutional networks, and doing this for classification tasks. We'll also learn how to fine-tune pre-trained models using a smaller data set and evaluate the impact of that on the model performance. So we'll have a hands-on session looking specifically at radiomics, or where you define a region of interest on the image. This is typically done in oncology, but it has been done in other applications also. And we then derive statistical metrics from these regions. We'll use open source packages such as PyRadiomics, and we look at some of the standardization of the radiomics application in the field of medical imaging. In terms of assessment then, it's split into 60%, which is 2 practical reports, so 30% each, one focusing on segmentation and the other on classification. And this will be done using open source data. And so students will split the groups fairly, assessing different expertise across groups. And the first one focusing on segmentation and the next on classification. And you will have the support of the tutors in starting off this process. You will have had lectures to that point so that you will understand the techniques, for example, in segmentation. We'll have covered UNet and some of the other segmentation models. But then it's up to you as a group to try and find some innovative approaches or ways that you can generate your results to do something new or unique about that. And the same with the classification tasks. The second assessment is 40%, and that's on individual presentations or rather recorded videos. So it's a special topic. You can choose any topic that's relevant to AI in medical imaging, something that you might be interested in or something you want to learn something more about, and that will be pre-recorded and submitted. So that's all for me, just to give you a bit about my own research, and I'm happy to take questions on the module also. So the research that I do is quite broad in terms of medical imaging and diagnostics. So some work is on digital twins, developing multi-scale, data-driven models for cardiac, specifically cardiac pathology, hypertrophic cardiomyopathy, other work in oncology, differential diagnosis, a lot of explainability work. So many different applications in medical imaging. So thank you for your attention and welcome to take questions.